So yes, I'm today the woman in red because I'm on a stop signal. Stop, danger, pathogens are coming. So I'm going to talk a little bit about the cannabis pathogens. As you see, we can have a very healthy and happy plant and a very sad one. And why is sad? Because many kind of pathogens could attack this plant. I'm not going to talk about pests like aphids, flies, mites, spiders, and so on. I want to talk today about these small ones that sometimes you don't see until it's too late, okay? So to have a disease in a plant, is the interaction of those three factors: the plant, the strain. Some strains are more resistant than others, the age of the plant, also the health of the plant, of course, the pathogen, fungi, bacteria, viruses, viroids, and the environment. It's so important that you control completely your environment because it's one of the factors that are going to influence the appearance of a disease, like the moisture, temperature, stress, vectors like insects, and more. So I want to talk first of the big or kind of those microorganisms, fungi. What are fungi? The first time that I listened about fungi when I was studying at the school, I said, no plants and no animals? What's this? This is a very stupid thing. And believe me, it's like they have their own kingdom. So it's like the kingdom of the ring. It's something so strange. They are eukaryotics. They have, I don't know if I have here some kind, yes. Oh, their own nucleo with the genetic material inside, but they need a host to live, and they can reproduce sexually or asexually, normally by spores. That is a very big problem because those spores are so resistant. So you think that you clean your plant, but the spore could be there. So harmful fungi species can cause serious problems for cannabis plants and can infect and cause diseases leading to reduced growth, yield, and low quality of the final product. Are considered harmful are the ones that rot, decompose, and damage the cannabis plant. And they can appear in seed, leaves, roots, stems, and flowers everywhere. Okay, of course, in soil, in air, so on. So, fungi pathogens of seeds, we have here Rhizopus, oryzae, and alternaria that may cause this one. I don't know what is this. Okay, poor seed germination and dumping off of younger seedlings, and it's the same for alternaria. But the most important is the transmission through seed is an important method for pathogen dissemination. But we have also fungi pathogens of roots, Fusarium, Phytium, Rhizotocnia. All of them mainly produce crown rot, root rot, dumping of diseases in plants, including cannabis, and could provoke stem cancer. Of leaves, this is very well known for everyone, powdery mildew, that suddenly you started to have this kind of white, I don't know, where is this? Okay, of white spots on the leaves and also on the flowers, and you are going to lose all your crop production because you are not going to have high quality flowers, okay? And this, this is one of my favorites. Why? Because Botrytis cinerea. Botrytis loves to live inside the flower. Oh, I don't know where is this. Inside the flower, because it loves to live very hot and with very high humidity. So it's just in the middle of the flower. When proliferates a lot, you lost all your crop production, you see something like that. But for me, this is not the very big problem. The very big problem is when it's still inside, it's like in the Caribbean, it's very happy with this humidity and this hot temperature. You harvest your crop, dry your crop, process your crop, and when you are going to validate your batches, suddenly in the middle still is botrytis. So you are going to lose all your batch production. What happens with bacteria? Bacteria are prokaryotes. This means that they don't have nucleus. The DNA are, of course, the DNA are inside the bacteria, but without a nucleus. There are prokaryota, and there are so many. You have all the kind of bacteria that you can have. And they could provoke many kind of symptoms. But I need to say that bacteria are not so harmful like fungi or like viruses and viroids, and are more easy to control than the other kind of pathogens. Now, start with my favorite ones, viruses. But what is that? Are not alive, are only DNA covered with a protein coat, and they could not live without any host cell, and suddenly they could go inside your cells and make many kinds of diseases. They are considered not alive organisms, okay? They are, oh, sorry, I'm going out. Okay. Plant viruses can be transmitted by an assortment of vectors through contact with an infected plant sap, via living organisms, for example, insects and nematodes, and through pollen. At the point when plant viruses are transferred between various plants, this is known as horizontal transmission. And when they are inherited from a parent, this is known as vertical transmission. And now we are going to see the most important for cannabis, okay? We have the lettuchlorosis virus, first time described in letuc, that you start to have this kind of yellow leaves with a stunted growth, so and also a reduction of trichomes, and can be asymptomatic for weeks. Okay. Tobacco mosaic virus. Many growers think that they have tobacco mosaic virus. Since this moment, there are not in the literature any positive test of tobacco mosaic virus in cannabis, but I'm very sure that it's going to be in cannabis for sure. So it's very important to test for this virus. This virus is very curious because you can see here that have different kind of patterns in the leaves that you see that perhaps is tobacco mosaic virus, but not much evidence for symptoms. Tests often come back negative. Similar symptoms to other problems. May common issues can cause similar symptoms, including incorrect pH, watering problems, root problems, deficiencies, etc. And could be genetics. Two-tone leaves variegations is a common mutation, so nothing worry about if plants are otherwise healthy and fast-growing. Other viruses. Cannabis cryptivirus is a very strange virus, but normally it doesn't express if it's not together with another one. And bit curly top virus. That causes the leaves to cool on themselves and alters the photosynthetic capabilities of the plant, which leads to less growth and less yield. And now this is the star of the party, the big problem that is nowadays all around the world, that is Hop latent viroid, the smallest and the worst. But which is that? This is not a virus, it's only RNA with anything more, and could be latent in your crop for many weeks, all your crop production. Also during many generations. It's like, I think that in English is like a squatter, occupy in Spanish, that is one person that has nothing, come into your home and started to use your washing machine, your TV, your computer, everything, and he has nothing, and normally destroy your home and go to another home. This is Hop latent viroid for cannabis industry. So the big problem is could be in your crop by generations and you don't know it. So you think that you have a healthy plant, make your clones, put your beautiful crop, and suddenly when you are going to harvest your crop, you are losing everything. Why? Those are the big symptoms. I need to say that those pictures that you are going to see about Hop latent viroid symptoms are coming from a very nice presentation of Dr. Junta that have some very impressive ones, okay? The first symptom is stunted growth. Then a poor development of the flowers. Also a very poor development of the root system. So your plant could fall down, okay? And those are, for me, the most impressive ones. Reduce seedlet development, making stems weaker. So again, your plants could fall down. And this is the more impressive, the reduction of trichomes. 50% reduction of cannabinoid production. Look, a very nice trichomes compared with symptomatic systems. That no terpenes or extremely reduction of terpenes content. And of course, reduction in yield. So this is a resume of all the possible pathogens that you have in cannabis. On bold letter are the most problematic ones, okay? And how to avoid, prevent or control all those pathogens. This comes from 2Me Genomics that makes a very nice white paper about how to avoid those pathogens. And there are many things that we need to do in this industry, okay? First, protect your plants from people. Please, not only workers, also visitors. Don't wear the same clothes that you have outside your facilities. You need to come into your facility clean. Continuously sterilize surface and equipment. Okay, it's very important also to say that normally, since many years, everyone clean with ethanol or isopropylic acid or something like that, but viruses and viroids are resistant to that. So please, 10% of solution of bleach, not ethanol, okay? Sterilize planting media. Normally cocoa comes with fungus and another kind of diseases. So it's important to sterilize planting media. Remove plant debris promptly. Every leaf, everything that is fall down on your facility is a place that is for the pathogens a palace to live, okay? Remove sick plants carefully. Don't take the plant and go running for your building, trying to keep out. Sterilize your cutting tools and hands between each plant. I know that is too much work, but you need to do it, okay? Of course, testing regularly. You need to test your plants to know if they are infectious or not. Control the flow of work in your facility. Normally, the cleanest part is your mother room, because are your genetics, are the most important of your company. And the worst part is the flowering room, because normally in flowering period is when all those pathogens attack. So control the flow of work in your facility. If you are in the flowering room, please don't come back to the other places, okay? Group clones together according to the mother plant. Why? Because if one mother plant is infected, you are going to know exactly what clones are infecting coming from this mother plant. Minimize pathogen spread with cutting clones. More or less, we talk about that. Important, please replace mother plants frequently. Mother plants are getting older, and if are getting older, they are going to be less resistant to all these kind of pathogens. Maintain adequate distance between plants. Plants could not be together, tossing one to another. Observe your plants regularly. Test all new material upon entry. If you are importing genetics or something like that, you need to test your material enter to go to the production model. Request evidence of pathogen testing for those people that brings you material, or if you could do it in-house. Ensure adequate filtration of air and water. And at least install insect screens. Those are some things that you can do at your facilities, but now we are going to see how really we can avoid those pathogens. So the solution to clean your plants, meristem in vitro culture. For some of you that perhaps you don't know how is in vitro culture, in vitro culture is something like the traditional cutting system, but made in a sterilized condition, okay? So you have, So you have a plant, and you excise a plant under sterilized conditions that you put in a growing medium, and it's going to grow and grow and grow several times, as time as you want. After that, you make the rooting in vitro and acclimatate and flowering the plant coming from in vitro. Tissue culture have 5 main advantages. remove pathogens from contaminated strains, avoid genetic drift and degradation of strains, ensure disease-free plants, achieve standardization and homogeneity, and increase flower and cannabinoid yield. This is all the types of in vitro culture that you could have. You could have sexual progeny if you make in vitro culture with spores, seeds, or embryos, or asexual if you make meristem culture, node culture, or tissue cell culture. Also, it could be divided into somatic embryogenesis if you induce an undifferentiated mass of cells that are going to produce a somatic embryo, or organogenesis that could be also indirect by callus or direct by microsuits. And after that, you are going to make your in vitro routine and your acclimatization. I don't recommend this part and also this part because normally have a somaclonal variation, which means that for breeding purposes could be nice because you could have a spontaneous mutation, but for propagation is not nice, okay? Why we can avoid the pathogens of our plants? Only by this, by using meristem culture. And why? Because meristem culture is the tip that is in the apical part of the plant in the axillar tips of each leaf that are completely aseptic because the flow of the plant doesn't arrive to this tip still. So if you have some virus or viroids, this is completely sterile by the moment, okay? Without pathogens. Sometimes it's not like it's not like this, and you need to make pretreatments of the plant or also the meristems. Heat, UV light, use virucides in your culture medium, something like that, okay? But you take those apical dome, put on in vitro, and each one is going to give you a very nice plant, virus-free. These are photos from our facilities. This is a meristem when you isolate the meristem, and then 3-4 days after growing, and 1 week after growing. And then this plant, you could grow the plant and have a virus-free and pathogen-free plant, okay? So how is the workflow on all of this? You have a candidate plant, and before nothing, you need to make virus testing and know if your plant are healthy or not. If you are lucky and your plant are healthy, you're going to have the varietal integrity verification by molecular markers to know that your variety is yours and not for others. You make your initiation in vitro, your propagation in vitro, test again, and if it's negative, you make the routine in vitro, and finally you have your plant clean stock that could go for make clean mother plant stock or ready to flower tissue culture plants. What happens when it's positive? When it's positive, you are going to discard the plants completely if you have not a tissue culture, or eliminate the pathogens with proprietary pathogen elimination protocols of tissue culture, and then again you make your virus testing, and if it's negative, again you are going to have your clean mother plant stock and you're ready to flower tissue culture plants. How you are going to make this testing? By molecular biology, by PCR. Nowadays, after COVID pandemic, everyone knows what is a PCR. PCR is not more than detecting the nucleic acids that are into your sample. So you amplify the number to have the possibility to detect if it's present or not something else that your own plant. So you could have endpoint PCR, that is like that only indicates the presence or absence of the pathogens nucleic acids, or real-time PCR. Not only indicates the presence, also quantify the quantity. viral load or microbial garden. Very important for your validation of batches when you need to have a viral load and microbial garden under some limits. So this is the positive control to know if your plant is infected. So if you are lucky and your plant is not infected, you are going to have something like that. But if the hop latent viruses are present, you are going to have this other kind of cures indicating the quantity of virus that are into your samples. So the process is like that: meristem treatment, isolation and regeneration, then multiplicate and make segment initiation of this clean mother plant. You make the rooting and acclimatation of those nice in vitro plantlets, but also in vitro culture is very nice and important because you could also make slow growth and cool preservation, and you could store as your stock genetic material for many, many months, clean and ready to go into production. So finally, let me introduce you a little bit about our company. We are MIFCO Biosciences. We are a Spanish licensed company based in Antequera, Málaga. Here is our CEO and founder, Antonio, that is today with us. José Luis Valencia, co-founder and responsible of infrastructures, and myself, Veronica, co-founder and responsible of cultivation. And our core purpose is to help medical cannabis cultivators and breeders by guaranteeing their biosecurity, protecting their genetic material, and maximizing their operations. Our mission to be the biggest European cannabis nursery by providing high-tech tissue culture services to breeders and cultivators, and our services, unique and exclusive genetics for the European market, full supply of Gene Zero clones from tissue culture rooted clones to ready-to-flower plants, clean mother stock supply to maximize clone production, ensure cloning success and keeping healthy plants, cleansing and rejuvenation program to eliminate all kinds of pathogens and avoid genetic drift caused by aging, and long-term genetic preservation. So please don't wait to have a problem, avoid it since the beginning. Thanks.
Thank you very much, Veronica. It was amazing. Are there any questions on the floor? We have got 10 minutes before we have to introduce our next speaker. So we could do one question if there are any questions on the floor. I've got a question. Veronica, you mentioned that the plants shouldn't be touching each other when they're in their growing cycle. At what point of the plant's growth cycle do you have to separate your plants? Because you can see in multiplication, for example, they are housed in the same sort of multiplication box. When do you sort of cross that threshold of needing to keep the plants separate?
I think that I know that it's very difficult to attain that, because you need to maximize your space, but it's very important since the beginning. Normally, let me tell you, they depend on the pathogen, because some pathogens like Fusarium or Oidium, that today I didn't talk about Oidium, are by the spores are very common to transfer from one to another, and if one leaf is just with another leaf like that, it's going to produce molds and so on. So the perfect is since the beginning, or at least ensure that your density is the appropriate one, depending on your strain. If you tell me how many plants per square meter, depending of how you strain looks. If it's more sativa, perhaps you can put more. If it's more indica, you need less plants per square meter. Also, you can make pruning of the leaves since the beginning. Also, the basal ones to protect this part. And if you could not make that, at least you need to ensure the correct air ventilations that could move this plant, and if this plant is together with another, but it's not always touching, it's with an airflow, a correct airflow.
I've got another question. It's not related to that. Speaking to legality, are you operating under a research permit, or do you? Because I understand in Spain there's quite tight regulation.
Yeah, yeah. We have since just 2 years ago an R&D license, but we are now constructing our big lab facilities. So we are asking for the production license to start operations at the end of this year, beginning of the next year.
That's very impressive. Thank you very much, Veronica. Thank you, guys.
You're welcome. Me saque esto.